Welcome to part 2 of 3 on chapter 6. We are looking at simple patterns of inheritance. In the first part of this chapter, we looked at Mendel's two laws of inheritance, so his law of segregation and his law of independent assortment, to look at how one trait and then two or more traits gets passed on from parent to offspring. In this section of the chapter, we're going to be looking very quickly at the chromosome theory of inheritance. So this theory really pulls together chapter 15 and this chapter, chapter 16, into this chromosome theory. So this is a theory on how genetic material or how our traits get passed down from parent to offspring in relation to what we know about chromosomes and what we know about meiosis and what we know about Mendel's two laws. So there are 5 parts to this chromosome theory of inheritance. The first part that we learned about actually in the previous unit is that chromosomes contain DNA. We also learned that DNA is the genetic material that codes for all of the proteins that give us our traits and characteristics, and that genes are found on these chromosomes, and genes are those discrete units of heredity material that's going to be passed on from your parent to your offspring. So this part we definitely already know. The second part has to do with the 2 types of cell division. So one of them, we know that chromosomes are replicated and passed down from parent to offspring, and we know this occurs in meiosis, which we went through in chapter 15. Chromosomes are also passed on from cell to cell during the development of a multicellular organism. And this is growth and development of an organism, so this will be mitosis. So we know that these chromosomes, they're split up, they're pulled apart, they're passed on to daughter cells, either during mitosis or meiosis. The third part states that in the nucleus of a diploid cell, we have 2 sets of chromosomes. And these chromosomes are found in homologous pairs. So remember, we also talked about homologous chromosomes. Homologous chromosomes have the same size, the same shape, they carry similar genes. And now we know those similar genes can have variant forms on them called alleles. And those alleles code for different forms of that characteristic that we're looking at. And then Mendel's 2 laws are the 4th part of this chromosome theory of inheritance, and this also relates back to meiosis, so the formation of the gametes. So at meiosis, one member of each chromosome pair segregates or separates into one daughter nucleus, and its homologous chromosome separates into the other daughter nucleus, and this relates back to that law of segregation. So our two alleles in our genotype, they segregate or separate from each other during this gamete formation. And again, this happens during meiosis 1. And then during the formation of the haploid cells, the members of different chromosome pairs segregate independently of each other. So you get this really wide range of variety that can end up in your gametes. And that relates back to Mendel's law of independent assortment. And then the 5th one, this has to do with the formation of the next generation. So these gametes, they're haploid cells that we just formed, so the eggs and the sperm, they combine to form a diploid cell during fertilization. And I know we talked about this in the previous chapter as well. So with each gamete transmitting 1 set of chromosome to the offspring. So we get 1 chromosome in the sperm, 1 in the egg. They come together and you get your zygote, which now is back up to a diploid chromosome number, so it has 2 copies of every chromosome in it. And you can see that in the image. So the haploid sperm has 3 chromosomes, the haploid egg has 3. They come together, you end up with 6 chromosomes or 3 pairs of homologous chromosomes in your next generation in the zygote. So that is the chromosome theory of inheritance. And just to kind of go into a little bit more detail so you really understand that this all relates back together. So Mendel's law of segregation can be explained by the pairing and segregation of homologous chromosomes during meiosis, specifically meiosis 1. So now we're going to be looking at chromosomes, and we're going to be focusing on chromosomes that have a certain gene on them, and they have alleles. Okay, so here we're just starting off with a mother cell, a mother diploid cell that just has 2 chromosomes or 1 pair of chromosomes to make it simple. So one chromosome has a little t on it, the other chromosome has a big T on it. So what happens is you go through meiosis 1. So your chromosomes get replicated in S phase, and now they look like the little X's. So each chromosome now has 2 sister chromatids on it. So we have our red chromosome and our blue chromosome. Those chromosomes line up in their bivalent. That bivalent gets attached to each of your 2 poles, and remember this is meiosis 1, so we have bivalents lining up in the middle during metaphase. Those 2 homologous chromosomes get pulled apart, so the one chromosome in the pair, the red one, goes to the left, the other chromosome in the pair gets pulled to the right. So now we have 2 haploid cells. They each have 1 chromosome in them, instead of a cell that had 2 chromosomes, which we start off with at the top. So we have 2 haploid cells at the bottom. One of the cells just has big T's, and the other cell has little t's in it. Then those 2 cells go through meiosis 2, and you end up with 2 cells that have little t's and 2 cells that have big T's in them. So we end up with our 4 haploid cells. So this really again shows Mendel's first law of segregation in relation to what happens during meiosis. Mendel's second law, his law of independent assortment, can also be explained by what we see during meiosis. So now, instead of 1 pair of chromosomes, we have 2 pairs. So we have 2 big chromosomes that have Y's on them, and 2 little chromosomes that have R's on them. And then we have another cell on the right-hand side that has the same setup. And we have 2 cells because we're going to show how we can get a bunch of different variation within our gametes. So those cells, they go through the S phase of interphase, so the chromosomes are replicated. So now we have chromosomes that have 2 sister chromatids on them. Your homologous chromosomes line up in their bivalent pairs, so our 2 large chromosomes are right next to each other, the 2 small chromosomes are next to each other. During metaphase 1, those chromosomes can line up in different ways. So if you look at the cell on the left-hand side, we have a large red and a small blue on the left, a large blue and a small red on the right-hand side of the cell. If you compare that to the cell on the right-hand side, we have 2 blues on the left, 2 reds on the right. So you can see these chromosomes, or the bivalents, can line up in lots of different ways, which can cause all this genetic variation we see with Mendel's law of independent assortment. Those homologous pairs get pulled apart, so we get the cells shown at the bottom, and then those cells go through meiosis 2 to pull the sister chromatid apart, and we end up with all different sorts of combinations of our chromosomes. So if you look on the left-hand side, we have a big R with a little y, big R, little y, little r, big Y, little r, big Y, big R, big Y, big R, big Y, little r, little y, little r, little y. So we get all 4 different combinations. And this goes back to that FOIL method that you do in order to determine the gametes that are being produced by the parent. So again, these 2 laws really relate back to how chromosomes behave during meiosis. So again, these ideas, they really go together, and they're not separate. So really think about it in this bigger picture and how genetics works with meiosis. So it's pretty neat stuff. Alright, so we went over this chromosome theory of inheritance. Remember, it's a theory, so there's lots of evidence to support it. Hopefully those 5 things we went through we already know because we've seen all that information in previous chapters. This is just kind of pulling all of these ideas together to see how they relate to each other.